Okay, thanks so much for joining, everyone. We have another great journal club today um, with Ryan. He is a student from University of Pretoria, biochemistry student to be specific. He has experience in applied enzymology for bio-waste valorization. I'm not sure what that term means. And the characterization of polysaccharide extracts using analytical chemistry, biochemistry, and spectral techniques. So XRD, FTIR, and my favorite NMR. He's a member of the Eucalyptus and Pine pathogen interactions group, which specializes in investigating plant disease interactions and plant defense in forest trees. This knowledge is then used to develop markers for disease resistance, which in turn are used to breed resilient trees in use for commercial forests in South Africa. He is part of a group which is working together with commercial forestry companies such as SAPPI and Mondi. He investigates the chemical composition of leaves from different eucalyptus trees. So he is a eucalyptus expert and determines if either resistant or susceptible to a fungal pathogen, specifically Chrysoporthe austroafricana. So I'll probably butcher that name a little bit. So he aims to identify specific compounds that associate with resistance to the disease from this pathogen. And he wants to find any metabolites that serve as markers to assist in breeding eucalyptus trees that can resist this fungus. So this is far outside my area of expertise. I'm hoping there'll be some individuals in the audience that will have some good questions later on, but I'm going to then pass it on to Ryan to give his presentation. So the floor is yours, Ryan.
Thank you very much, Prof. Just to answer your question, bio-waste valorization is just basically converting bio-waste into useful products, either medically or economically. It's what I worked on in my honors year. So it's a bit of a far cry from what I'm doing now, but it is something that I do actually feel very passionate about.
Well, I've learned
But,
something already.
anyway, thanks for the very nice introduction. I'd just like to touch a bit more on that. So EPI works with various forestry industries. I mentioned SAPPI and Mondi because they're our biggest, but actually there's, I think, a host of over 30 companies we do work with. And we are very integrated with them and their own research departments. And our group mostly focuses on genetic markers for various things like disease resistance or cold tolerance, drought tolerance, those sorts of things in these forest trees. So mine is a bit of a new project for the group because it's the first time we're actually looking at the metabolomic side of things. But to get started, some background on our subject. So, as I mentioned, eucalyptus plantations are important to South Africa. Forestry in general provides quite a lot of revenue for South Africa and many jobs as well. And eucalyptus, behind pine, is the second most planted commercial tree in South Africa. We produce, millions of tons of timber from these trees. And one specific one, Eucalyptus grandis, is very important because of various properties that are good, like its rapid growth. The wood is very nice for pulping. The leaf oil contains medically relevant compounds. And this tree is so desirable that even though it's native to Australia, it's planted worldwide. However, it is susceptible to diseases like Chrysoporthe austroafricana. So Chrysoporthe is a canker disease. It forms these cankers on the stems of the plant. It's in this specific species is believed to be native to South Africa and underwent a host shift from our species onto eucalyptus, where it causes disease. And it forms these little black fruiting structures with orange exudates. And it is responsible for quite a bit of mortality. It was a big disease a few decades ago. And even though it was managed with breeding trials, there is some evidence that it's popping up again in field. And it is a big focus of research in plant pathology. In fact, it's like a well-described pathosystem, the Eucalyptus Chrysoporthe pathosystem. There's a lot of research going on in this system because of how relevant it was in the past. So how this generally works, these breeding trials that I mentioned, is that we would inoculate bunches of trees with this pathogen, and then you'd let the disease develop and then see how far it develops and use that to infer whether a tree is resistant or not. So the trees that were planted that managed to deal with this in the past were hybrids of Eucalyptus grandis and Eucalyptus europhylla. And it worked very well. But like I said, if it's going to come back, it's going to be a bit of an issue. And the inoculation trial has issues in that it is very sensitive to external factors. It is slow. You have to wait for the trees to grow up quite a bit before you can do them. And that makes them expensive because a lot these trees go to waste essentially. So it's not really desirable. So I've just got some photos of some of the work that we've done, us doing these inoculation trials in greenhouse, and some of the lesions and cankers in field. So to sort of deal with those issues of inoculation trials, we want biomarkers of resistance. So as I mentioned, our group focuses on genetic ones, but I'm looking at metabolite biomarkers, which are small biomolecule indicators of the state of an organism. And they can serve as a proxy measure of the phenotype. In this case, the phenotype would be that lesion length that we see. And they can provide information on the state of the subject. The one relevant to us is whether the tree is resistant or susceptible, which means it can then be used in resistance screening, and that is desirable for breeding programs. But additionally, looking at these chemicals, it can help us identify the mechanisms of defense, which is very important for plant pathology research, because we want to know which pathways and biomolecules are involved in defense, because that has various applications downstream. And it just adds to the knowledge base of the pathosystem, so other researchers can benefit from it as well. So my project started with some past research. These are two students in our group. Well, I guess the one's now a postdoc. But they looked at, they had a little bit of an insight into the chemical nature of resistance. And they saw that the terpenoid levels and genes surrounding the terpenoid expression are differentially induced in our tree against Chrysoporthe. So that sort of gave us the idea that, hey, maybe we should have a look at these terpenoids, right? Another research groups in FABI, which is one of the groups I'm part of, is doing a similar project on insect pathogens of eucalyptus. And they actually found some pretty nice biomarkers with these three at the top here. When they're low, we see that this insect attacks them more, and when the bottom two are there, they attack less. So quite promising. We hope to find something similar where we can now just look for these chemicals in addition to genetic markers instead of having to run whole inoculation trials. But outside of our group, similar studies have been done on trees in the same family and other eucalyptus trees as well, and also finding evidence of some of these terpenoids being important for resistance to either fungal pathogens, but also insect pathogens. So this brings me to the aim. So I want to find these metabolites or signals that are associated with resistance to Chrysoporthe. And we have some questions about that. So we want to know, okay, which ones correlate with them and what is their role in eucalyptus biology? What do these terpenoids do? When are they activated, kind of thing? And then a bit more practical, how well can they be used to distinguish resistant profiles? Will they actually be valuable in breeding trials or not? That is sort of the questions we want to answer with my project. So basically the objectives is firstly, we need to find out which clones are resistant and susceptible using lesion data from inoculation trials. Select those clones, extract metabolites from them, and then get spectral data. Then we want to annotate the chemicals. We want to see what exactly they are. And then we want to see how well can they be used to identify resistant clones in other trials. So the clone selection starts with analyzing the lesion data, identifying resistant and susceptible clones and selecting them. These will then, we will gather tissue from these clones, extract their metabolites, and then perform spectral analysis. The main ones we're focusing on is LCMS-MS, derivatized GCMS for the polar compounds, and then GCMS for the volatiles. And then we'd pre-process the data, identify and characterize ones that could be biomarkers, and then interpret the results, see what these chemicals are, what role they play, and how well they can be used as biomarkers. So to give you a bit of idea of how we do this clone selection, we have 200 sort of standard genotypes from industry that were generated through a cross between Eucalyptus grandis, Urophila, and then backcrossed to the grandis parent. And we've been doing greenhouse inoculation trials since 2020 on these plants. And we have quite a bit of lesion data from that. But last year was quite nice. We started with our first field trials, and we did one in Kwambanambi, which is a bit of a warmer, hotter region, and then Richmond, which is not as warm. And the reason for this is we want to sort of see, do these things hold up? Do these biomarkers hold up in different locations or under different climates, to put it that way? Because it wouldn't really be that helpful if the biomarkers can only be applied under a certain condition or in certain regions, considering that these trees are planted all over KZN and there's quite a varying climate. So we would take the lesion data and we would just identify those that differ significantly from each other. And the ones with smaller lesion lengths, we'd say they are resistant, and the ones with the larger lesion lengths, they are susceptible. We selected 20 from each that we were confident were resistant and susceptible. And we had to sort of decide, are we going to take a few clones and do a lot of reps of one of those clones, or take a lot of clones instead of, and have less reps? So we went for a lot of clones because we're working with populations of trees. If whatever biomarker we only find in a certain few trees and it doesn't, and we don't find it in the rest of the population, once again, it doesn't become useful because then it's limited to just those few genotypes. So that was our reasoning for that. So we did tissue harvest from the field, from the field trees. We wanted fully open, undamaged leaves at a consistent location on the plant. So it was the north side and 1.8 meters above the ground. We wrapped them in foil and then immediately put them between bricks of dry ice to sort of maintain the metabolic profile. And then later on, when we got back to the lab, we could store it at -18. Then we did our metabolite extraction. It was done with Morton pestle and a liquid nitrogen. And then we did our extraction with either methanol for the polar compounds or hexane for the volatiles, using 2-diphenyl ethanol as a standard. Separated the insoluble material out by centrifugation, and then stored it at -20 degrees Celsius. And then we wanted to send some for GCMS and some for LCMS-MS. And then we wanted to derivatize some of the methanol extracts so that we could also run it on GCMS. So both of those would be the polar compounds, just run with 2 different techniques. However, there were a lot of delays in this, unfortunately. Our GCMS was dowered for about 6 months, so I only just got that data and didn't get to look at it. In the meantime, we actually ran some samples on just normal LCMS for some, just to have a look at what's going on. This is just the workflow for how I did it. For the methanol extracts, I first lyophilized the tissue. We didn't do that for the GCMS samples because that can get rid of a lot of the volatile compounds, which is what we wanted to measure. So that we had to do fresh. We'd weigh it, do our extraction, and then pellet out the insoluble material, and then we could store the supernatant with all our metabolites. So I mentioned that LCMS analysis we did. So we had about 1,200 features, actually bins. I didn't separate them, or I didn't do deconvolution yet on them, but just to see what was going on. And we found that of that, 118 of them were significant. So how I did this was I actually used a lot of different techniques, but I'm just showing the results from a logistic regression that I did on that. And at the 5% level, we had 118 of them that were significant, but none of them were, or only 3 of them were actually good at predicting on their own. And I also did a heat map of those compounds so we can see sort of the difference between the groups. And the ones in red is sort of the starker contrasting images where we can see how they differ between the 2 groups, resistant or susceptible. And the next step was to sort of annotate these features. So the LCMS, we couldn't really use the data in for database searching. So we had to sort of look at past literature where people did similar studies and try to see if they found metabolite features that were similar to ours. And we want to compare with the Richmond data, which is the next field trial we're harvesting in May. We want to see if those patterns sort of match up. And additionally, we want to look at the actual LCMS and GCMS data, which I've started with now. I wish I could show that, but unfortunately, yeah, we did have those delays. I just also want to acknowledge my supervisors and co-supervisors, my team, and also my funding bodies. Thank you very much.
Thanks so much, Ryan. It was a very interesting study you're doing. Lots of people are involved, so I think you're playing a very important part there. So good job so far. I'm going to open the floor to the audience. If there are any questions, you can either raise your hand or you can type in the chat box. While we wait for questions, I have maybe it's a stupid question, or I don't know if you consider it, with your experimental design, because from what I understand, the pathogen, the fungal pathogen attacks the bark, the stems of the or the trunk of the tree. Why don't you analyze that part? Why are you analyzing the leaves?
That is a very good question. It was something we considered early on in the project. It does make more sense to look at the bark, but we wanted to look at the leaves because that way we can do non-destructive sampling of the trees. When we take bark samples, we open up the trees to further infection. So ideally, we would want to be able to just take leaf samples and see if we can find those patterns. And what we also sort of expect is that when we look at this plant pathology, a plant that is resistant to similar types of pathogens that may be infecting the leaf as well, a lot of the same biological pathways are involved. So we would sort of expect that even though we're not looking in necessarily the locale where we expect our pathogen to be, if it is resistant to similar pathogens, we expect that to be a sort of systemic thing. So we should still find those markers elsewhere on the plant.
Okay. I know Prof. Gerard is our resident expert. There he is with his hand. So please, Prof. Gerard, ask your question.
Yes, thanks for the presentation. I was just looking at some of those molecules, compounds, and it seems like some of them might be volatiles or very volatile. I don't know if I'm right, but you are not thinking of looking at the volatile um, compounds that's around the tree, and how that is influencing the resistance or not.
Let me just make sure I understand. Are you saying not the volatiles from the tree, volatiles that surround the tree? Um,
Yeah, yeah. So they call it the VOCs, the
yes,
volatiles
yes,
around
yes.
the tree that's released from the tree.
Yes,
So yeah.
I think that could be certainly an interesting avenue of research. Maybe it's something we do look into in future because you are right. The volatiles do seem to be important. It wasn't the focus of my project, but yeah, I think that would be something interesting to look at. As I mentioned, there's also a lot of students working, on similar, or in a similar area. And I do know for some of the insect pests, they do look at the volatiles and they have found that, yeah, they are very important. So yeah, that is certainly something to look at in future.
Okay, thanks, Prof. Gerard. I know myself and Prof. Gerard are NMR guys, so I'm going to ask the question, why haven't you done any NMR yet?
So initially we wanted to do NMR, but we sort of had to decide, are we going to do NMR or LCM-SMS? And we sort of selected LCM-SMS. We didn't have the funds to do both necessarily. I also wanted to do NMR. I think it's quite nice as well. But we
Well,
just thought
NMR
that,
is much cheaper. You see a lot less, but with higher confidence.
yes, exactly. The confidence there is nice. Um, I did look at quotes, at least at our university, and the LCM SMS actually ended up cheaper. So I don't
Okay.
know.
Well, I'm biased, so I'm the NMR guy at NWU, so I'm obviously going to push NMR for any project.
Yes, yes. We were considering sending NMR samples to you, actually. I think we spoke a bit about that last year. We were trying to get the design right for that, but
Not
in
not
the
not
end,
to not to me. Don't send it to me. Send it to Prof. Gerard or Fadal at UJ. They're the guys who do plant metabolomics. I don't have the databases to identify the compounds.
I see. But I believe Proper Postleaders has sent you T samples in the past.
Yeah, he sent me T samples and 20, 30 pure compounds to create a small database specifically for the Ts. So if you accompany the samples with some of the ideas of the compounds you're looking for, like you saw now, you've got 4 or 5 suspects there to look at. You want to measure those suspects in your samples, I can do that, no problem.
I see. Okay. Well, if the project continues into PhD, then maybe that can be an option, or we can send it to Prof. Harrod as well. I suppose we'll have to decide then.
Well, there you go, Prof. Gerard. There's potential collaboration in the future. Um,
I, yes. Sorry, I might have missed. Um, I probably missed it. This pathogen you're looking at, is it found? It's is there a vector that's carrying around? So how does it infect?
Yes. So, there's not a specific vector, no, but it does release spores, and when there's open wounds on the trunk of the tree, that is where they sort of enter. And from the natural growth of eucalyptus, you often find that cracks form at the base. Um, so that does make them quite susceptible. Because yeah, that just happens as part of their natural growth. There's not even necessarily something damaging them, something external damaging them. But yeah, so that's how they enter.
Ryan, can you just go to your very first slide? Okay. Yes, so if anyone wants to contact this group for questions or ideas involving collaboration with the project, so there are the supervisors. Ryan, can you just put in the chat box your email address if anyone wants to contact you? So if anyone watching on the YouTube channel, because this is being recorded, I'm going to upload it to join all the others there. If anyone views it afterwards, then they can then contact you. I'm sure you'll be open to any inputs or discussions regarding this. So the email for Ryan, for those that can't see it in YouTube land, is the name ryan.bosch@up.ac.za. So I'm sure Ryan will be open to any emails regarding his project.
Yes, I would like to discuss, because I'm still learning, I won't lie. There's a lot of questions I have about the best way to go about doing these things. So yeah, discussion is certainly welcome.
Good. So I hope you do get some communication. If there aren't any other questions from the group, I think we can end it there. I wish you all